So I mentioned before with the MAO pathway, one of the most critical components of Harmine, one of its most potent targets, is the DYRK1A pathway. Okay, and DYRK stands for dual specificity tyrosine. Tyrosine is abbreviated with a K. That's where that K comes from. Tyrosine regulated kinase. Okay, I think I got that wrong. Dual specificity. Tyrosine is with a Y. Sorry, tyrosine is with a Y. Regulated kinase, DYRK. And then there are a couple different subsets of the DYRK pathways, 1A, there's 1B, there's 1C, I believe, as well. But don't get lost in the sauce. Actually, when I read DYRK1A, I say in my head, DYRK. So, if I'm reading a paper that reads that out, I don't read out DYRK1 every single time. I just say DYRK, the DYRK pathway. So this particular pathway, the DYRK1 pathway, involves enzymes that correlate with cell proliferation and brain development. Okay, so again, ties to neurological disorders. We talked about Parkinson's before. We talked about the psychedelic and the spiritual component of ayahuasca. It has a lot of ties to disease, and when you think about dealing with cell proliferation, whether that be an overgrowth of cells, which we think of with tumors and cancer, or an undergrowth of cells like we think of with beta cells and diabetes, it makes sense that this would be influential in the health of cells. And sure enough, I've got a paper here titled The Omnipresence of DYRK in Human Cells. So this just goes on to say that DYRK does so many things to not be important, right? And if there is any sort of dysregulation in DYRK activation or inactivation, you're going to feel it, right, in the body. So DYRK is a subclass of protein kinases. I think I've mentioned this before in podcasts previously, but protein kinases are like on-off switches in the body, right? These kinases are things that go around and turn proteins on when they're needed, turns proteins off when they're not. This is just one specific case of protein kinases, the DYRK. So DYRK has multiple biological functions, okay? Cellular signaling, mRNA splicing, chromatin transcription, DNA damage repair, cell survival, cell cycle control, neuronal development and functions, and synaptic plasticity. I mean, this is like an all-star protein kinase, okay? So cellular signaling, we're interested in insulin resistance. We're interested in beta cells knowing when to grow and when not to grow. mRNA splicing and chromatin transcription, DNA damage repair, this has everything to do with the creation of proteins. It has everything to do with the regeneration of proteins, healing damaged DNA in states of oxidative stress, which inherently ties into cell survival. So this is like such an important component of a healthy cell cycle. It's definitely something that we need to be paying attention to, and it's not surprising at all that targeting this is something that would benefit those with type 1. So Harmine is seen as something that gets beta cells to start to proliferate, which is something that is not easily done, or so we're told. So Harmine inhibits the DYRK pathway, right? So it's inhibiting the inhibitor. DYRK is something that stops beta cells from proliferating. By stopping what's stopping proliferation, you're actually promoting more beta cells. So in that initial study that I mentioned at the beginning of the podcast, in each of the different origin styles of diabetes, whether it be pancreas is physically damaged, whether it is immunologically damaged, or whether it is inflammatorily damaged, right? Either it's cut in half and you lose part of your pancreas, the immune system isn't able to protect it, or there's just so much oxidative stress going on in the pancreas that it ends up dying and the beta cells stop. Harmine and its effect on the DYRK pathway was able to induce growth in all three of these models. Of note, it was not specific to beta cells. It was able to grow beta cells, but it grew a lot of other things as well. And alpha cells was one of those things in the pancreas. And I've also mentioned this before, talking about how diabetes isn't just regrowing beta cells, it's restoring the balance of alpha and beta cells. So this is a little bit of an obstacle when it comes to actually treating type 1 diabetes, because it's growing beta cells isn't enough. It's a matter of also stopping the growth of alpha cells to allow for beta cells to kind of close the gap and restore balance between the two. Good news, though, last bullet there, no observed DNA damage or cell disruption. So this wasn't something that was like, if you use too much, the cell goes kaboom and it's worse. You're worse off for using it than you were not at all. So this is something that has therapeutic potential.